So identifying cell surface receptors for viral entry is critical to understanding virus infection. And that's because gaining entry into a cell is one of the most important molecular hurdles for a viral particle, since failed entry is complete dead end for infection. Put another way, an invading army can't claim a medieval fortress if it can't scale the castle walls. Despite this, we often don't know some of the finer details about receptor-virus interactions. For example, we all know that ACE2 is an incredibly important cell surface receptor for the SARS-CoV and SARS-CoV-2 spikes, where once the spike protein binds ACE2, it can then be activated by cell surface proteases like TMPRSS2, or it can get endocytosed and cleaved by endosomal proteases like cathepsin L to trigger fusion. The virus is now in the cytoplasm and free to start replicating. Well, beta coronaviruses have specialized receptor binding domains, which I'll refer to as RBDs, that dictate the affinity and specificity of the interaction. The RBDs for SARS-CoV and SARS-CoV-2 are about 220 amino acids and is the main determinant for spike binding with ACE2. Well, as far as we can tell, also baculovirus RBDs have very similar tertiary structure, even when the primary sequences are quite different. For example, here's an overlay of the SARS-CoV and SARS-CoV-2 RBDs, and they look this in sync despite sharing only 75% amino acid identity. Well, the clade 1 baculoviruses, including SARS-CoV and SARS-CoV-2, are generally found in rhinolophus bats in or around China. But these aren't the only clade of SARS-like coronaviruses that exist. And there are also clade 2 viruses, which are also generally found in and around China. But these viruses have been found in other bats, including Rhinolophus pharyngium bats that have a range that extends across Asia into Europe. And the story gets far more interesting with clade 3 SARS-like coronaviruses, which have been found in a bunch of diverse horseshoe bats in Western Asia, Europe, and Africa. We know that SARS-CoV and SARS-CoV-2, like clade 1 viruses, use ACE2, but what about the two other more distantly related clades? Well, here I'm showing you an identity matrix where you can see how many amino acid differences there are between two RBDs, where white means there are very few amino acid differences, while black means there are greater than 75 differences. And as you can tell, even by eye, the clade 3 viruses seem to exhibit at least some similarity to the clade 1 RBDs, while the clade 2 was consistently different from both of these other clade RBDs by approximately a third of all of its residues. Well, a couple of previous studies tested a couple dozen clade 2 RBDs and a few clade 3 RBDs, and the investigators didn't observe any ACE2-dependent enhancement to infection. Thus, it was concluded that neither clade 2 nor clade 3 sarbecoviruses use ACE2 as the entry receptor. But there are some caveats when trying to interpret negative data. For example, it's possible that there were mismatched sequence compatibilities, as some of these virus RBDs may only have affinity for specific ACE2 orthologs from their host species that weren't tested yet. It's also possible that the RBDs have weak affinity for ACE2 that's hard to measure with conventional assays. And along those lines, ACE2 can really vary in sequence, particularly in horseshoe bats, which are thought to be the natural reservoirs for these viruses. For example, ACE2 orthologs from various horseshoe bats routinely differ by 50 or more amino acids. And even some alleles within the same species, like the Chinese horseshoe bats of Rhinolophus sinicus, can differ by up to 17 amino acids. Thus, we set out on a series of experiments to try to better understand these receptor requirements for these more diverse clades of SARS-like coronaviruses. And to actually test this, we turn to lentiviral pseudotypes. Here, the viral particles are coated with viral entry proteins that we want to test, and successful entry allows the lentiviral vector to insert a GFP cassette into the cell genome and mark that cell as infected. But we also created two different stable cell lines, one expressing human ACE2 at a high level, and another cell line expressing just the cytoplasmic tail of ACE2 to serve as a negative control. I've colored the nuclei of these cells different colors, since they each express a different fluorescent protein as a reporter. But that also means it's possible to distinguish the two cells based on their fluorescence profiles, so we don't need to keep them in two separate wells and can instead mix them into the same well being infected. Thus, when they are challenged with a GFP pseudotype virus, we can use GFP to figure out if the ACE2 expressing cells were more infectable than the control. And we essentially do that using flow cytometry, which is used as a high-speed cell counter, where we can separate the two cells based on their red or near infrared fluorescence. And then within each of those populations, figure out how many of those cells became infected by looking for green cells. Here's an example of what that data looks like, where VSVG uses LDLR as a receptor, and pseudotype viruses with VSVG infected both cell lines equally, while in contrast, viruses pseudotyped with SARS-CoV spike were 15 times more likely to infect ACE2 overexpressing cells rather than controls. So to demonstrate the specificity of this approach, we dug into our freezers and found plasmids encoding viral entry glycoproteins from a panel of different viruses, some of which use different receptor proteins. And as intended, only the SARS-like coronavirus spikes using ACE2 gave clearly enhanced infection in cells overexpressing ACE2 as compared to cells that weren't doing that. And with that assay seemingly working, we tested a panel of horseshoe bat ACE2 orthologs along with human ACE2 for comparison. And each of these orthologs are shown on this heat map as columns of the x-axis. When we exposed each of these cell mixtures to pseudotype viruses coated with SARS-CoV chimeric spikes, encoding our panel of diverse RBDs, and these RBDs as shown are shown on the heat map as rows along the y-axis. Here I'm showing results with our two negative control samples, neither of which yielded any effect. While the clade 1 virus RBDs, including SARS-CoV and SARS-CoV-2, exhibited broad ortholog compatibility, including human ACE2, which was consistent with a bunch of existing literature. The clade 2 virus RBDs didn't show any clear ACE2 usage, including with the orthologs from the bat species they were isolated from, which are signified by the black rectangular boxes. The clade 3 RBDs were more interesting, as half of them were incredibly specific and only using a single ortholog that we tested, while the RBD from Costa 2, found in Sochi, Russia, was similarly specific but could also use human ACE2, while the RBD from Costa 1, also from Russia, more broadly used various orthologs but could not use human ACE2, and BTKY72, found in Africa, had an RBD that was both broad and capable of using human ACE2. Seeing the distinct patterns of ACE2 ortholog usage was interesting, but the orthologs differ at multiple positions, making it hard to narrow down key residues responsible for the phenotypic differences. We thus decided to revisit a panel of human ACE2 mutants that we had previously tested in a prior publication and see how Costa 2 and BTKY72 spikes fared when infecting this mutant panel of human ACE2. Well, the most interesting results were with the K31D mutation, since we observed contrasting effects depending on the RBD. For example, Costa 2 really did not like aspartic acid at this position, whereas BTKY72 did. And we hypothesized that compatibility of the side chain at ACE2 position 31 may depend on the identity of the corresponding nearby amino acid on the RBD. And looking at known structures, this corresponded to an amino acid extending from a highly conserved beta sheet right at that interface. Well, this position is quite variable depending on the RBD, and is asparagine or glutamine in SARS-CoV or SARS-CoV-2, as well as Costa 2. While in contrast, BTKY72 and Costa 1 both encoded lysines. And digging further into that ACE2 ortholog data, both Costa 1 and BTKY72 seem to disfavor ACE2 orthologs encoding a K at position 31. And this led us to hypothesize that RBDs encoding K at this position were likely generally poorly adapted to ACE2 orthologs that encode K at position 31, like human ACE2 does. And we decided to test this empirically with rationally designed mutant RBDs. Here I'm showing you the ACE2-dependent infections observed with wild-type RBD sequences. And we first checked to see what happens if we take compatible amino acids and change them into lysine. And we didn't see much of an effect for SARS-CoV or SARS-CoV-2, although we previously showed that these RBDs are so good at binding human ACE2 that it's hard to see effects from single amino acid mutations in ACE2 overexpressing pseudotype virus assays like ours. On the other hand, Costa 2 is presumably less well acted overall, with presumably lower binding affinity, and mutation from Q to K caused a greater than 10-fold decrease to infection. We also tested the opposite situation, where we mutated the lysine in COSTA1 and BTKY72 into glutamine or asparagine, and there was little effect with COSTA1, suggesting that this change was insufficient to enhance infection by itself, whereas BTKY72 did exhibit a tenfold increase to infection when mutated to glutamine. So we think this is indicative of a possible molecular route towards zoonosis, where an RBD like BTKY72, which already has some affinity for human ACE2, first acquires that lysine to glutamine change and render it far more capable of entering human cells. Amazingly, there's a precedence for something similar having happened with the original SARS-CoV outbreak. And there it is believed that a K-encoding precursor virus was replicating in palm civets and eventually acquired an asparagine mutation, which helped it jump into humans for that first 2002 outbreak. All that said, there's no need to panic since BTKY72 entry is still very inefficient, and there are also a bunch of other steps in the viral life cycle that likely need to be adapted for actual successful zoonosis. But while we should continue to watch for the clade-1 suspects, it could be worth also monitoring for clade-3 viruses on other continents like Africa. Doing so will likely reveal additional common themes in zoonosis, like we may have uncovered here in this project. But there's also more to be done. For example, what's the receptor for the clade-2 viruses? Hopefully somebody, not us, will figure that out. But we do plan on creating much larger compatibility data sets using a fully multiplexed sequencing-based format. And with such large data sets, that will aid our long-term goal of using these data sets to train algorithms capable of predicting compatibility based on host and virus sequences alone. With that, I'd like to thank the individuals who performed this work in my lab, as well as the collaborating Bruchet lab, as well as the NIH for support. Thank you.